these are your video notes for Chapter 11 on cell communication. Now, before we get started, I want to point out to you that cell communication is a very, very broad topic. It's not something that you can really cover in just one chapter. This is meant to be an introduction to overall how cell communication works. As we go through the year, we'll talk about lots of different types of cell communication. We'll talk about it in the nervous system, we'll talk about it in the immune system, we'll talk about it in homeostasis and feedback loops, and in hormones and how they work. There's lots of types of cell communication. We'll talk about it even when we get into gene regulation. There's lots and lots and lots of different ways that this works. This is just an overview as to how most cell communication works. So just kind of keep that in mind as we're going. The evolution of cell signaling. Signal transduction pathway is a received signal is converted to a specific cellular response in a series of steps. So what you need to get from this is a STP is sometimes how this is abbreviated. Is a signal transduction pathway. So it's a pathway for where we're inducing or sending a signal. So you're going to have a received signal will be converted to a response. That's what you're getting out of it. Similar in yeast and mammals, even though they are very different organisms. Similarities in bacteria and plant cell communication also indicate cell signaling mechanisms have evolved long ago. This shows some cell communication with some, looks like some spores, some mold here. Individual rod-shaped cells, they aggregate in progress, they form a structure, a fruiting body, and so all of these cells have to communicate with each other in order to get the end product created. Quorum sensing allows bacteria populations to coordinate behavior so they can carry out activities. Think about bacteria, they don't talk to each other, but they do send signals or chemical messengers to each other, and that's going to allow them to work together to create a common good, or a common, well, for them, maybe not for us, but to create some kind of common goal. That's called quorum sensing. You want to know what that is. Similar to long-distance signaling in many human cells. And we'll get into what long-distance signaling is in human cells. So there's two different basic signaling. Think about it kind of like telephones. You can do local or you can do long distance. So some cells are going to communicate by direct contact. One cell is actually in contact with another. There are small junctions between the cells that connect the cytoplasms of adjacent cells. It's called cell-to-cell recognition. This is really important in embryonic development and the immune response. In embryonic development, the way this is really important is the cells that are next to each other will communicate. They'll send signals across gaps or junctions, and that will tell the signal which genes to turn on and which to turn off so that it appropriately makes itself into a skin cell or a muscle cell or whatever it is. And you think about it, you start from one single cell, and that cell divides into 2, and that divides into 4, and that divides into 8. And so when you're thinking about this happening, these cells have to send signals, and they have to communicate with each other in order to know whether they're supposed to be a brain cell or a skin cell, so that ultimately what you get is you get this perfectly formed human body, or any other organism for that matter, that is perfectly positioned. And this just shows some pictures here. Gap junctions between animals. In between plants, it's called plasmodesmata. You want to know that term, plasmodesmata. It's basically just a gap junction in between 2 plant cells. And so cell recognition is going to be where the cells are really close together and they're recognizing each other.
Good morning, Washington High School. At this time, please release all seniors to the CJB. All seniors, please report to the CJB.
Sorry, we have an announcement. Okay, so there's going to be a some kind of a receptor protein here and one here, and they're going to recognize each other. So local regulators, messenger molecules are secreted by the signaling cell. They travel short distances. Think about local. Example, neurotransmitter in synaptic. I always struggle with that word, even when we get to nervous system. Synaptic signaling and growth factors in the pancreatic signaling. get some pictures here. So, so here is, for example, like in the pancreas, think about, I keep touching this. It's not. Think about here, like the cell is going to form some kind of a chemical into a vesicle, and it's going to secrete it, and then it's going to go hit a target cell. Usually that's still like a really short distance, something that doesn't need to travel very far. And then in a neuron, this is another really good example. Here's neuron 1, and this is neuron 2, and the we call neurotransmitters, because neuro for the nervous system is basically a chemical that is being transmitted from one neuron to the other. And this one's going to pick that up, and then it's going to send it down the axon of the next neuron. And that's how brain cells communicate with each other. Long distance signaling, think long distance. It's going to be, the signal is going to happen in one area, and it's going to travel a further distance to actually produce a response. The best example here would be hormones. Think about in you, if you're a boy or if you're a girl, you're going to produce hormones, either testosterone or estrogen, and actually you guys both produce a little bit of each. Males produce more testosterone, females produce more estrogen. And those hormones are going to travel throughout the body, even though they're produced in either the testes or the ovaries, they're going to travel throughout the body and produce the different effects. Like in males, the testosterone will produce the extra hair growth and the deep voice, those kind of things. And in females, that's going to produce like breast production and things like that. So those hormones are produced in one localized areas, but they're traveling throughout the body, that more long distance. And animal hormonal signaling is part of the endocrine system. So here's an example where the hormone travels in the bloodstream. So the hormone is packaged and it's excreted out of the cell. So this is an ovary cell. And it's going to get into the bloodstream, and then it's going to travel to whatever target cells it needs to go to, which may be more than one thing. So there's three parts of cell signaling, three main parts, and these look different depending on what the cell signal is or what the cell communication is. They vary cell to cell type and organism to organism, but they still have three main parts: reception, transduction, and response, RTR. Reception is the target cell's detection of a signaling molecule coming from the outside cell. The transduction is the converting the signal to a form that can bring about a specific cellular response. And the response is when it actually triggers a specific cellular response. So reception is the target cell's detection of the signal. Transduction is converting the signal, and response is actually producing some kind of cellular response to that signal. This is kind of a animation of it, or a model of it. I will tell you, this model appeared on the AP exam one year as a free response question. And I don't, sometimes they reuse those same images. They just use them in different places. They might take it and put it in a free response one year, and then one year they might put it in multiple choice, like that. But this one, very similar to this, was in there. So here's your cell. This is the membrane. That your signaling molecule is going to find a receptor. The cell is going to recognize it. It's going to change it. Okay, that's a transduction. It's going to convert that message. And usually this is a stepwise. Lots of different things can happen here. And then whatever the response is, that's going to be whatever. Okay. So reception, transduction, response. Remember these three, and remember what each of them do. Reception. A receptor protein must recognize the shape of the molecule it needs to recognize. The signaling molecule behaves as a ligand, a molecule that specifically binds to another one. The ligand binding causes the receptor protein to undergo a change in shape, which activates the receptor. Examples: G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. And I'll show you those. You don't have to know them specifically. This is a G-coupled receptor. Okay, it's called that because it has something called GDP coupled with it. So that's going to couple it, and that's going to activate that receptor. And then you're going to get the transduction, and then you'll ultimately get some kind of cellular response. This one is the tyrosine kinase. This is have these extra tyrosines there. And the signaling molecule is going to bind, and that's going to activate something to happen. And it's going to do a transduction, and then ultimately you'll end up with your response. Ion channels here again, signaling molecule is going to attach to it, and then you're going to have some kind of a flow in through the membrane. You can have that transduction, and ultimately you'll have a response, and then they get flow. The transduction part is multi-step pathway. That's really important to understand. One benefit of a multi-step pathway is that you can amplify the signal. Think about what amplify means. You can make it stronger. Okay, you can make it stronger. Another benefit is more opportunity for coordination and regulation in simpler systems. So it's fine-tuning that response. It's going to make it a little bit better. Signal transduction means that one receptor activates another molecule, which activates yet another molecule, and so on, until the final response is activated. Think about the activity we did in class. We'll do that if you haven't already by the time you watch this. Often these involve molecules changing shapes as the signal is passed. And this just shows an example. So you got the signaling molecule for reception, and then this whole thing here is going to be transduction. Okay, you have basically a step-by-step, different things are happening. In this case, it looks like we're adding some phosphorylation, we're adding some phosphate energy going on there too. And ultimately, you have your response. Sometimes a second messenger is essential. They're not proteins, and they're usually water soluble so that they can diffuse easily. Commonly used second messengers are cyclic AMP and calcium ions. Know those two. You'll see this one when we get to the neurons. And then cyclic AMP, I think we talk about, I can't remember what unit we talked about. Maybe we talked about it in this one. Some pathways include protein modifications, which regulate the transduction pathway. For example, adding phosphates or adding methyl groups. So phosphorylation or methylation, adding phosphates or adding methyl groups. So you've got your first signaling, so you've got your reception, and then here's your second messenger. That's going to help out the process, and then you've got your response. The response ultimately leads to the regulation of one or more cell activities. Response at the end of the pathway might be in the nucleus or it might be in the cytoplasm. Termination occurs when the number of active receptors falls below a threshold, which can happen through a variety of ways. Termination occurs when the number of active receptors falls below a threshold. So here's the growth factor. It's being received. You've got your transduction. You've got into the nucleus. Now we're doing something in our response here. We're actually transcribing a gene. That's what's happening here. You've got DNA and you're transcribing this gene. That's your response. It's actually creating the mRNA. Once that's done, then we can go back and it can turn that pathway off. You will research one defect and create a presentation about the cell communication that has gone wrong. We're going to talk about that in a second. Apoptosis. Cells that are infected, damaged, or have reached the end of their lifespan. Cells don't live forever. Okay, depending on the cell type in your body, depends on how long it lives. A skin cell might live a couple of weeks, a few days, whereas your brain cells are going to live most of your lifetime, if not all of your lifetime, unless you do drugs and damage them, in which case they're not coming back. The pathway that triggers apoptosis depends on the cell and the signal that initiates apoptosis. So what apoptosis actually is this programmed cell death. It's cell death that happens for a reason, because the cell's infected, it's damaged, or it's just done. Cancer can be caused from a defect in the apoptosis process. What cancer can lead to is it can lead to tumors. And tumors are just masses of cells that shouldn't be there. Excuse me. And so what can happen is that if there's a problem in the apoptosis process, the cells don't die. They don't stop dividing. They don't die. And when they don't die, you can end up with a mass. The cell is not getting the signal to die, and it's damaged. And then we have a problem. We have a damaged cell that's not dying. Now you have a problem. You have damaged cells infecting other cells. And doing that, they're sending more cell communication to other cells. Hey, I'm damaged, I'm bad, and I'm going to spread it to you. Apoptosis is also really important in embryo development. And you see here that you actually start with your hand is all kind of one solid piece, and then the cells in between your fingers actually apoptosis. They die. If you've ever seen anybody with like webbed feet or webbed fingers, it's not really a problem other than those cells in those areas just didn't get that signal to die. And so now they have that remaining tissue. Okay, that's it.